Hello everyone and welcome back to my another tutorial class on modes to speciation. The event or the process of genetic divergence of gene pool of populations to form new species. While talking on the mechanisms, several possible mechanisms are proposed to account for the occurrence of speciation. For example, Mayer 1963 and Endler 1977 commented on some possible modes like phyletic transformation, where speciation is with respect to time, the gradual replacement of older species by the new ones without branching in due course of time, or we may also refer to as anagenesis. Another possible mode is the fusion of species, in which a new species may arise by fusion of two already existing species through the breakdown of the reproductive isolation, which we also know as hybridization. The third mode, which is called the true speciation, here the speciation occurs due to splitting of species because of reproductive isolation. Today we are going to elaborate on this mode of speciation. The true speciation may be of two types: sudden speciation or quantum speciation, or also called as sympatric speciation. The sudden speciation or quantum speciation event occurs basically suddenly and intermittently. The main reasons for this sudden speciation event or mode is mainly due to the chromosomal aberrations like inversions, translocations, or change in the chromosome number, such as polyploidy, which includes autoploidy, alloploidy, which we shall discuss in more details in the following slides. On the other hand, gradual speciation is a microevolutionary event where small changes in the genetic composition grow with time, which of course may be favored by natural selection, thus giving rise to new species. The gradual speciation again may be of following types, such as allopatric speciation, peripatric speciation, and parapatric speciation. So let us understand the allopatric speciation. Allopatric speciation is the divergence of physically separate population of a species occupying separate geographical areas into new species. This is also sometimes called as geographical isolation. The way that leads to the physical isolation of populations may occur by three ways. One, the physical splitting of large habitat into small habitats by some new physical barriers. As you see in the picture here, a large parental population is dissected into two populations because of a physical barrier, which may be a river, an ocean, a glacier, earthquake, flooding, mountain, etc., which is often also called as vicariance. Here in the picture to the right is an example of speciation in warblers. Mengel 1964 presented with an example of allopatric speciation in northern wood warblers of North America. These warblers are represented by 4 species, 1 eastern and 3 western. The speciation event in these warblers was due to repeated glaciation event, which fragmented their larger habitat into isolated smaller habitats. The population of species occupying these isolated regions also got genetically isolated and evolved itself into new species. The second method leading to allopatric speciation or the physical isolation is by divergence of a few members of a widely distributed population to some new habitat, which we also call as founder's effect. And the third type, in the third type, there is an extinction of the intermediate links in a food chain of the interconnected populations, which may also result in the establishment of a new population. Peripatric speciation is a mode of speciation in which a new species is formed from an isolated peripheral population, which are called as marginal isolates. Peripatric speciation very much resembles allopatric speciation, but the difference of which lies in the size of the newer population. That is, one population is much smaller than the other one. Peripatry can be distinguished from allopatric speciation by three key features. The first one becoming the strong selection, which is caused by dispersal. Second, the colonization of a new environment free from competition. And the third one, the strong role of genetic drift, which works on the smaller populations. Smaller the population, stronger is the pressure of genetic drift or natural selection. Here in the example we see that the New Guinea paradise kingfisher bird Taniseptera galliata is widely distributed throughout the lowland, which is marked by 1, 2 and 3. But again, if you notice properly, it has differentiated into several distinct species on smaller islands, namely 4, 5, 6, 7 and 8, which are the marginal isolates. The third type of speciation, parapatric speciation, where para means near, is the development of a reproductive isolation among members of a continuous population or spatially distinct populations between which there is some gene flow in absence of geographical barrier. Basically, what happens here in this mode of speciation is that a population enters a new niche or habitat, but still remaining within the range of the parent population, which means the new put to be species is still in contact with the parent population, as a result of which there may be still a little but unequal gene flow between these populations. However, they would like to mate with the population of the geographical neighbor rather than the individuals of different part of the population range, which means the species will not travel to extreme boundaries just for the sake of mating. As throughout the large geographical range, there are varying amounts of selection pressures which act differently on different geographical range, which will slowly lead to the formation of a new species. Here in this simple diagram, you see the green is the parent population. Now the species starts to diverge against a cline. Here cline means a measurable gradient in a single character or a biological trait, which may be genetic, like the change in allelic frequency, the blood type, or maybe phenotypic, example the body size, skin pigmentation, etc. Now the divergence which is seen in the form of a blue color. The blue color is not totally a new species, but with a small difference along the cline. the gene flow may take place between the gene and the blue, but in a reduced form, because the species now would mate or try to mate with species within their own geographical neighbors. Meaning, the green will mate with green and blue will blue. Though green can mate with the blue. Now, as the original habitat range is big, the natural pressures of selection will be different in two extremes. Say, green is a cold environment and blue is a hot environment. As a result, the green will try its best to suit for the colder environment and the blue for the warmer environment. Although the species of blue and green will still interact along the edges or near the ecotone to form hybrids. Slowly with time, the accumulation of differences due to selective pressures will finally divide them into two separate species of green and blue, denoted here by two separate circles. The example here is of a green bulbul in the tropical forests of Cameroon, which occupies two habitats, that is grasslands and the adjoining forest. The overlap of these two ecosystems is a ecotone. The species living in these two different habitats, one being the grassland and other the forest, will soon be acted by different selective pressures in two different habitats, thereby leading to some phenotypic differences in the bulbul of both the habitats. However, the two species along the line will mate in some edges of the ecosystem, that is the ecotone, thereby forming a hybrid of both types. But the changes that have been introduced in gene pool is more than the actual gene flow occurring along the edges. The divergence in the gene pool overshadows the effect of gene flow between each of them, and each population thus establishes itself to becoming a new species. Till now, I have detailed you on the various modes of gradual way to speciation, but here on discussion is sympatric speciation, which comes under sudden or quantum speciation. You need to understand first the speciation event is not an event which occurs in a few days or years of time. It happens in terms of geographical timescales, where even a single period within an era is separated by millions of years. This is important to discuss here because although we describe sympatric speciation as to be a quantum or a sudden speciation, the speciation event does not occur in a matter of few days or years. So again, going back to the topic, sympatric speciation refers to the origin of new species due to appearance of some biological barrier in the individuals of an initially random mating population that live in the same geographical area. So, what are the characteristics for this mode of speciation? Firstly, sympatric species occur in the same geographical area. Secondly, they are not separated by any physical barriers. And thirdly, as there is no physical barrier, there is a steady gene flow amongst the members of the population. Lastly, the populations, though may be with same geographical area, but they may be isolated by preferences for different habitats and needs. So again, what are the mechanisms for a sympatric speciation? As I have already mentioned earlier, that as there is no physical barriers, there is a steady gene flow amongst the members. However, sympatric speciation occurs by genetic divergence in the gene pool between members of the population. So, the important question is what causes the reduction of gene flow to result in sympatric speciation? The reduced gene flow is a consequence of ecological isolation or habitat differentiation and disruptive selection. With the large geographical area, each population wants to stabilize itself in the new habitat or in the new ecological niche. Henceforth, disruptive selection occurs within the larger population. So again, now what causes the biological barrier with the population? Within the population, the answer is polypolarization or change in chromosome number and chromosomal aberrations, and also introgressive hybridization. Introgressive hybridization means gene flow from the gene pool of one species to the gene pool of another by repeated back crossing of a interspecific hybrid with one of its parent species. I shall discuss it in more details in the later half of this presentation. One classic example of ecological isolation leading to sympatric speciation is of North American apple maggot fly. As the name suggests, North American apple maggot flies, the one pictured below, can feed and mate in apple trees. The original host, however, for these flies is hawthorn tree. It was only when the European settlers introduced apples around 200 years ago, some flies were adventurous enough to exploit apple as a food source. The flies that were born in apple trees tended to feed and mate with other flies in the apple trees, while the hawthorn flies fed and mated in the hawthorn trees. Over time, the population diverged into genetically distinct groups with adaptations. The host shift from hawthorn to apples may be the first step to sympatric speciation in fewer than 200 years. Some genetic differences between the group of flies evolved were established, though some interbreeding still occurs between the apple specialized flies and the hawthorn specialized flies, so they are not separated into species yet. However, some scientists think this is a case of sympatric speciation in progress. Again, the second cause leading to reproductive isolation, that is of course leading to sympatric speciation, is polyploidy. In this condition, an organism, due to improper meiosis or mitosis or meiotic or mitotic error, will result in more than two sets of chromosomes, where if 3 sets it shall be called triploid, or if it is of 4 sets will be called as tetraploid. These generally occurs as a result of total non-disjunction of chromosomes during meiosis or mitosis. The condition is the main reason of speciation in plants, especially in the case of angiosperms. Unlike humans and other animals, plants are often tolerant to these changes in the number of chromosome sets. As you see in the schematic below, a diploid plant, due to the total non-disjunction of chromosomes during meiosis, will produce diploid gametes, which in turn of haploid gametes, 2n in case of n. Again, these gametes fuse to form a tetraploid organism or a plant. Here in this slide you see in the schematic above the same case I just discussed before, and in the lower schematic two species, species A with chromosome number 4 and species B with chromosome number 6. Again, in species A, error occurs during meiosis, resulting in reduced gametes, whereas meiosis occurs normally in species B, resulting in normal gamete. These gametes from species A and B hybridize to form a new type with uneven chromosome number of 7. These hybrids will again backcross with the normal gametes from the normal parent B, again producing a viable and fertile hybrid of even chromosome number, that is 10. I have put a simple example of a onion to make you understand polyploidy and speciation. Here you see the normal onion with a diploid set of 16 chromosomes, allium cepa. Now, as a result of polyploidy, the autoploidy, an error in the meiosis led to producing a diploid gamete which fused to form a polyploid, that is tetraploid 4n with 32 chromosomes, double the number of chromosomes as seen in allium cepa. This polyploid onion is a new species called allium tubersum, or also called as Chinese tribes. So this sums up the various modes of speciation. Thank you for your patient hearing.